we'll be starting off with the revision of the various signaling pathways that we have, right? So let's begin. This is one of the most important unit. Reason being, you get very straightforward questions from here. Okay, very straightforward questions. First and second, every year from the GPCR or from the calcium calmodulin or the cholera toxin, in some way or the other, questions are like more or less guaranteed from this particular segment. Okay, so try to have a very good grip over the various sequence of the pathway, like the complete sequence of the steps that occurs in every signaling pathway. That is very important, right? Is it clear to all? Right, let's begin with the class now. So, talking about this particular unit, we have already covered the cell adhesion molecules and we have already covered the cancer topic. Now, we are starting off with the signaling pathways, right? Talking about the various signaling pathways, before that, we need to learn about what communication is all about, right? That what is communication. So, in simple terms, what do you mean by communication? At present we are having a form of communication only, right? When I am trying to send a message and you are trying to receive a message via the choice of words that I am using, right? Via the words that I'm using. So I am the sender, you are the receiver, and the message is sent via the words, via the language, right? So same goes with the cells as well, because the cells cannot have a verbal dialogue, so they use signals to communicate with each other. So what is cell communication or what is cell signaling? It's a type of communication that is occurring or that is happening between two cells, so that they can they can complete a specific task in a specific manner, right? Because, like, if we talk about the thing that we discussed yesterday, that is the two cells being in contact with each other, how do these two cells come in contact with each other, or how do these two cells signal that we have to stay over here? This is via what? This is via signaling, right? Now, based upon the same, based upon the signaling type, like based upon the signaling, we have majorly four types of pathways of signaling. Can you write down in the chat section which are the four pathways of signaling, or which are the four types of signaling? Write down, everyone. Which are the four types of signaling that we study in the communication? Think about it quickly. Yes, which are the four pathways? We have stacrine and paracrine, right? What do we have? We have autocrine, we have paracrine, we have juxtacrine, and we have endocrine. What are the four types? Like, what is the meaning of each one of them? Autocrine is when a cell is giving signal to itself. Auto means self. When a cell is giving signal to itself. For example, your pathway of in apoptosis, we see the self or the autocrine signaling. Paracrine is when the cell is in the near vicinity, when the cell is present in the nearby location, we see the paracrine signaling happening. Then we have juxtacrine, when there is a signaling pathway, when there is a physical contact between the two cells, that is what juxtacrine is. What is endocrine? Endocrine is when you have a cell which is releasing some kind of signals, and these signals are transported to the other cell via the medium, via your bloodstream, when the two cells are present in like when the two cells are far away from each other. This is what endocrine signaling is. So autocrine, paracrine, endocrine, and your juxtacrine. Remember about all four of them, because you can get a question in the match the following type, right? I hope it's clear. Next, so see over here, what happens to this? Just a second. Complete written part comes carry forwards to the next class. Next slide. I got it checked also so many times. There's no problem in the laptop. Just got a new one. Okay, see over here. So you have what is signaling all about. So you have the receptor, you have the transduction, you have the response. Receptor is like the reception is the one which is giving the signaling molecule. Signaling begins when the signaling molecule known as the ligand, it is binding to the receptor on the surface, and then the information is carried forwarded. Then you have transduction, that is the passage of information inside the cell, and then you have the response occurring, right? Four type of signaling: endo, para, auto, and juxt. I've just given you the information. This is the elaborated version for the same. I'm expecting all the pointers are clear to each one of you, along with the examples, because you'll get examples also asked in the examination. Okay, so make sure that you remember all of these. Now, talking about the ligands, write down what all are the various nature of the ligands possible. Yes, what are the various nature of the ligands possible which can help as a targeted sequences? Yes, think about it, everyone. Which can act as a target, or which can act as a signal carrying molecule? Think about it, everyone. Very good. Hormones, growth factor, transcription factors. Very good, Ayush. What else? What other factors could be there? Very good, Akansha. Amino acids, enzymes, hormones. So anything which can carry the information can act as a factor, can act as a ligand. See over here, small molecules could be there. You can have peptides, you can have the hormones, you can have lipid based ligands, you can have nucleotides, steroid, gases, etc. In gases, the signaling pathway that we study is for your nitric oxide signaling, which is very, very important, because in 2023 also there was a question asked from the NO signaling, right? Talking about the type of ligands you've just understood. In addition, like, you can keep in mind the broader level or the broader group of the nature: hydrophilic molecules, lipophilic, steroid, non-steroid, etc. right? Remember all these things. Now we have the various type of receptors. So receptor could be an ion channel receptor, it could be an enzyme linked receptor, it could be orphan receptors. Orphan receptors are what? The receptors which are still under research, and or if you want to learn it in a more specified manner, those receptors which have not, like, whose ligands have not yet been classified, whose ligands have not yet been discovered, right? So this is what orphan receptors are. Talking about the iron receptors, write down the most common example that we study for the iron receptors, everyone. Yes, iron receptors, most common example. One of the pathways there in which we have an iron receptor working in, which is it? That a specific ligand binds to it and then those iron receptors open up. Which pathway? Yes, it is IP3. IP3, after getting detached from the plasma membrane, reaches to which organelle? Yes, which organelle do we have in the IP3 pathway, from where we have the ion channels opening up, the calcium ion channels opening up? To which organelle does it goes? Yes, think about it. Smooth endoplasmic reticulum. Yes, it was smooth endoplasmic reticulum, on to which our calcium, like IP3 DAC, IP3 binds in. As a result of the same, you have the calcium 2 plus ion channels opening up, and as a result of the calcium ion 2 plus channels opening up, you have the calcium getting coming out of your SER, and then it binds with your kelmodulin. Then the further process continues, right? We have learned about all these things. Now, ion channel, then we have the enzyme linked receptors also, in which you have the utilization of the enzyme as a function, or for opening up some of the receptors, or for the functionality of the receptors, right? So examples are, there are a number of examples. Most important one is your RTK, receptor tyrosine kinase pathway, right? In addition, we have receptor guanylate cyclase, we have receptor serine threonine kinases as well, right? All these things we are going to discuss just now. Now, talking about the various signaling pathways, today let's target out your GPCR, IP3, cholera toxin, and your nitric oxide. Okay, let's target out these four pathways, and then we'll see according to the time. Okay, let's begin with the GPCR pathway. Talking about the GPCR, GPCR stands for your G protein coupled receptor, right? What are the various characteristic features that we have to remember for the same? It is a transmembrane receptor in which you have the which spans around the plasma membrane 7 times, right? You have the receptor spanning the plasma membrane 7 times, right? First thing. Second thing, you have the N terminal, you have the C terminal. We have the G protein initially in the inactive state, where you have G alpha, beta, and gamma, all these three in organized together, right? Alpha, beta, and gamma. Sorry, gamma, beta, alpha. These are the three subunits. Now what happens? Now you have, as and when we have the ligand binding to the receptor, that is over here, as and when the ligand binds to the receptor, we have the G protein getting converted into the active state. For the conversion into the active state, what is required? We have the G beta and alpha getting dissociated, and G beta and gamma getting dissociated, and the G alpha subunits become activated, and then the further pathway continues. Over here you have the utilization of guanine exchange factors, which help in the conversion of GDP into GTP, right? As a result of the same, what do we have? We have the G alpha getting activated, or your G protein with the alpha subunit getting activated, and then our further pathway continues. What is the further pathway for the same? The further pathway for the same is, you'll be having the activation of your, yes, which particular pointer get activated over here? Think about it, everyone. Yes, what's the next step over here? G alpha will be working at what step? Think. Which particular enzyme gets activated? It's a revision class, so you have to be very much active. Yes, adenylate cyclase. It will be activating what? It will be activating your adenylate cyclase. Adenylate cyclase will be helping in the conversion of ATP into CAMP. Ayush, the G alpha subunit will be activating your adenylate cyclase. Adenylate cyclase in turn will be activating, or will be converting your ATP into CAMP. Now, four of the subunits of your CAMP will be activating the PKA. What is PKA? Protein kinase A. From the inactive state to the active state, it gets converted into the active state. Now all these things are occurring in your cytoplasm. Now this PKA enters into what? It enters into the nucleus, where it will be activating your CREB. What is CREB? CAMP element, or CAMP response element binding protein. Right now, this will be in addition or in combination with your CRB, that is CREB binding protein. These two will be activating your transcription factors, and as a result, we'll be having the response in relation to what ligand was there will be showcased, right? So, this is your entire GPCR pathway. See over here, look at this particular diagram. This is your GPCR's like structure, the structure of the protein, and this is your entire pathway. Whatever I've drawn there, exact same thing is over here. So take a minute, go through all of this. If you find any difficulty, you can ask, then I'll be carrying forward. 1 minute for this, everyone. Done, everyone? Right. For the GPCR. And remember, I'm just making you revise all the pointers. You'll have to work hard to like keep them in your mind. Okay, just don't sit with this that 1 hour of four signaling pathways and I'm done with it, and I'm not going to open up the class again. No, please do not do this kind of stupidity and work hard. These are just a glimpse of the pathways, these are just the glimpse of the topics that we have already covered. Okay, now GPCR can also have a wide range of ligands, and based upon the ligands, you'll be having the similar responses seen, right? You can have neurotransmitters, you can have hormones, you can have odorants, chemokines, you can have lipids, light sensitive molecules, taste molecules, etc. Now talking about the structure of your PKA. What is PKA? Protein kinase A. Talking about the structure of the same, so you have two of the regulatory subunits, you have two of the catalytic subunits, right? As in when your cyclic AMP binds to it, you have now this catalytic the cyclic AMP binds to the regulatory subunit. As a result of the binding to the regulatory subunit, there occurs a change in the morphological structure of your catalytic subunit, and as a result of the morphological change in the catalytic subunit, they get detached and then the further pathway continues. Because this exact thing has been asked in the examination, that's why I'm insisting upon this. You have two regulatory and two catalytic subunit. Right now, CAMP binds to the regulatory subunit. And every regulatory subunit have two sites for the CAMP binding. As a result of binding to the regulatory subunit, there occurs a morphological change in the catalytic subunit, and as a result of change in the catalytic subunit, you have the activation of the PKA, and thus the further work continues. It's a very important and a very minor information to remember, but very potential statement as well. Okay, remember about this particular thing, everyone.